Hello.
Yes,
Okay, Professor.
yes.
Okay. So that
Can
means
you
you
hear
can
me?
hear us. Yes, I can hear you now.
Okay. You can hear me and see me, right?
Yes.
Yes, yes.
Oh,
Loud and clear.
um, okay, it's better. Okay, please, everybody, mute yourself. Mute all. I can do that. Okay. Okay, we'll wait until it's 4:05 and we'll start. Whew. Oh,
Bye.
okay, let's start. So I hope everybody understood that there's 4 days that you have to come to Hunter for the class. And I'm going to send an announcement with those 4 days. Every other class, it's going to be online. Just the exams are going to be in person. So I hope everybody understood that. It's very important. Okay, so last class, or the first class actually, we talked about all these systems that make up our body, the 11 systems, and we're going to study them separately because it's easier, but they all have to be together, work together to maintain this condition, which is homeostasis. Homeostasis is the same as healthy. People that are healthy are under homeostatic condition. And why I'm showing this, so this figure is balancing between stress conditions that disturb homeostasis and replies or conditions that will overcome that disturbancy. So you see here the components of this system that works to maintain homeostasis. So here you see a receptor. There's many, many receptors throughout our body, inside and on the skin, that can perceive changes in the environment. When you think about environment, you cannot just think the environment that is around us. That's the external environment. You have to think also the internal environment, where there's changes in oxygen, in carbon dioxide, and things like this that will also disturb homeostasis. So you have the receptors that are essential to perceive that there has been a change, and change in temperature, a change in temperature, a change in pressure, a change in light, and so on and so forth. So these receptors are specific for each condition, and they send a message to the control center. To let the control center know that there has been a change, that has been a disturbance in homeostasis. And then the control center sends a message to the effectors. So the effectors are the components of the body, such as the muscles or glands, that will respond to the messages from the control center. And then the effector tries to overcome the disturbance that caused by different stimuli. So this goes around and around in a way that it works to maintain homeostasis. So it's basically what we call a feedback loop, because the action starts in the receptor. When there's a change, the receptor perceives it, and then this mechanism occurs that is going to overcome the change that occurs. It's going to overcome the disturbance in homeostasis. So if you see me looking to the right, it's because I have to allow the students to come in the waiting room. So I'm sorry that I'm always looking at the upper right corner. Okay, so this is all very interesting. And this is what you're going to learn. How is it that the body can, with all the changes that occur around us and inside us, maintain these homeostatic conditions? Just think about the temperature. Our body temperature is normally 37 degrees centigrade or 98.6 Fahrenheit. And if it goes up to 37.5, just a half of a point, then this whole system initiates so to bring the temperature down to homeostatic conditions. So it's very important that we understand what is involved in all this circle that maintains the healthy individual. Okay, now homeostasis again. Now remember, or maybe you don't know, but in our body, there's around 100 trillion cells. 100 trillion cells. Just to give you the concept of what does it mean, 100 trillion cells. Look at this big, big tree. This big tree is estimated to have around 175 to 225,000 leaves. And each leaf on this tree needs to get the proper water, the proper nutrients, the proper oxygen, carbon dioxide. It has to be able to get rid of toxins. So each of these leaves on the tree is regulated, highly regulated. Now, this is only, let's say, 225,000 leaves. Now think of 100 trillion cells in our body, and each cell has to be maintained under homeostatic conditions. And the cells vary. A muscle cell is very different from a skin cell, from a nerve cell, from a cardiomyocyte, which are cells from the heart. So all these cells have to be maintained under optimal conditions. So what is it? What is necessary to be maintained at optimal conditions for each cell? They need to receive proper concentrations of gases, so oxygen, carbon dioxide, nutrients, and water. So if the cells are dehydrated, that means they're not healthy. The cells have to be maintained at an optimal body temperature, which I already said, it's around 37 degrees. And also the optimal volume. You don't want the cells, if they swell, they will burst. Like the red blood cells in the blood, it's very important that they maintain their shape and that they don't burst. If they burst, there's not enough. The red blood cells, their main aim or function is to carry oxygen throughout the body. If they're burst, then there's not enough red blood cells to carry oxygen, and this could lead to death. So the size of the cells, to maintain the optimal size and volume of the cells, is very important. I don't see there's one person. Okay, so let's move on. So let's say this is the body, a very simplified way of representing the body. Inside you have cells, and then outside you have the external environment. And you know that the external environment varies a lot. You can go from a very hot room to a very cold, if there's the air condition is on, or with this light to dark, or a room or a condition where there's more oxygen. And the body has to be able to overcome these big changes in the external environment. So the internal environment also varies, although there's smaller fluctuations. So there's changes in pH, in glucose, in oxygen, in carbon dioxide. So all these changes have to be overcome to maintain homeostasis. So you want to maintain a stable internal environment. That's the definition of homeostasis. So you have to be able to overcome large changes that occur in the environment around us. So in our body, there's different types of fluids. ICF is the intracellular fluid, ICF, intracellular fluid. That's the fluid that is inside the cells. And these fluids have different properties. So inside the cell, you have the ICF, which is the intracellular fluid. And then outside the cells, so out here, so this is the cell, the fluid inside the cell is the ICF, intracellular fluid, and then there's the fluid outside the cell, and that's the ECL, or extracellular fluid. So there's three types of extracellular fluid. Interstitial fluid is the fluid that bathes the cells, and it's not contained in any vases. That's the interstitial fluid. Then we have the plasma, that's also fluid, and that fluid runs through the blood vessels. It's extracellular because it's outside the cells. And then you have the lymph. The lymph flows through lymphatic vessels. So these three fluids are extracellular, they're ECF. So there's a lot of crosstalk between the ICF and the ECF. Let's look here. So these are cells. Anyone has a question?
Yes, sorry, I have a question.
Yes.
I didn't catch what you said about the intracellular, the one for the ECF, the first one.
That's the intracellular fluid, the fluid that is
No.
inside the cells.
The ECF.
The ICF. The ECF is extracellular. ICF intracellular. ECF extracellular. And there's
I'm talking about the
three
explanation for
different
the
types of extracellular fluid. Is that clear?
Yeah, I was talking about the explanation for the A, the one for the ECF, the interstitial.
This one.
The A.
Oh, A, interstitial fluid. I see. I'm sorry, I didn't understand.
It's okay.
The interstitial fluid is a fluid inside where it's not contained in vessels, so it's not in blood vessels or lymphatic vessels, but it bathes the cells. So the cells are in this fluid known as the interstitial fluid. So here you see cells with a nucleus, and the fluid inside is the intracellular fluid. This fluid outside the cells, that bathes the cells, is the interstitial fluid. So the interstitial fluid is not contained in vessels. Then you have the plasma that is in the blood vessels. So plasma is the liquid component of the blood. So the blood is made up of plasma and blood cells, the white blood cells, the red blood cells, and the platelets. But the fluid part, the liquid part of the blood is the plasma. And then you have the lymph. The lymph runs in the lymphatic vessels. So the interstitial fluid, the plasma, and the lymph are all extracellular fluids, meaning that they are not contained in the vessels. Okay, so and here you see another view of these fluids. So the yellow represents the lymph, so the fluid that goes through the lymphatic vessels, and these are very important in the immune system. Then you have the blood, the plasma that goes in, runs through the arteries and veins. The veins are here represented in blue, and the arteries in red. And then you have the interstitial fluid. So this pink here that is not contained in vessels is interstitial fluid. So what's the point of me telling you about these fluids? Because there's a crosstalk among all these fluids to maintain homeostasis. So, for example, you see here a blood vessel, and the blood vessel contains water, proteins, and ions such as sodium, potassium, and so on. And the walls of the blood vessels act as barriers. So water can go from the blood vessels to the interstitial fluid and into the cell, which would be the intracellular fluid. So water, because it's a very small molecule, and there's actually some proteins that form pores for water to be able to flow freely. These pores are known as aquaporins. So the water can flow from the blood vessels through the interstitial fluid and into the cell. But let's look at the protein. Proteins are large molecules, and they cannot leak out of the blood vessels. If there's leakage of proteins from the blood vessels into the interstitial fluid, that means that there's a problem with the walls of the blood vessels. So proteins are high or large molecules that cannot leak out of the blood vessels. Proteins cannot also leak out of the cells. So they are maintained either in the cells or in the blood vessels. Then you have these ions, sodium and potassium. Because they are charged, they're positively charged, the walls of the cells will repel them. They cannot go in freely. So sodium and potassium can leak out of the blood, but they cannot get into the cells freely. They have to go through special pores or channels. So all this is highly regulated. So this exchange, oh my gosh, I forgot to look at the chat. Is that the same fluid that prevents friction within the lungs? No, that's different. Now we are focusing on these. We're focusing on these fluids. The fluid in the lungs is different. It's protective. So we're not talking about this. We're talking about these fluids that are bathe cells, that are in cells and are in the blood vessels or lymphatic vessels. So this interchange between blood vessels, interstitial fluid, and cells is highly, highly regulated. You don't want to have too much You don't want to have too much sodium in the cells or too much sodium in the blood vessels. Can cause disruption of homeostasis, and you will learn why later. Okay, so again, homeostasis is a condition in which the body is healthy and it's able to maintain the internal environment. The internal environment is maintained under a very narrow physiological limit. The concentration of glucose, the concentration of oxygen, the concentration or the temperature, all these are maintained, all these parameters are maintained in very, very narrow range. And if the body cannot maintain homeostasis, then the individual is sick, disease occurs. So you could define disease as a condition in which the body cannot maintain homeostasis. Okay, so why is homeostasis disrupted? So there are all kinds of stress conditions that are known as stimuli that can disrupt homeostasis. Some are internal, some are external. So what are some of the external stimuli that can disrupt homeostasis? Changes in temperature, if it's too hot or too cold, loud noises, lack of atmospheric oxygen. So you know that in high mountains, the oxygen density of the air is much lower than at sea level. So when you go all of a sudden to a very high mountain, very high altitude, the decrease in oxygen density will cause, for example, headaches. Your body has to adapt going from an altitude where there's a certain amount of oxygen and then go to a very high altitude where there's less oxygen. The body has to adapt to that change. And how does it adapt? One of the factors is to increase the number of red blood cells. So I don't know if you know, but many athletes that run the marathon, they train at high altitude. And why is that? Because if they are at high altitude, there's a hormone in the body known as erythropoietin that increases the number of red blood cells. So at high altitude, this hormone is produced in higher amounts, therefore leading to an increase in red blood cells. And if it's an increase in red blood cells, then they are able to absorb more oxygen and therefore distribute the oxygen through the blood. If you have more of these red blood cells, it's very useful under conditions of high altitude. So if athletes train at high altitude and then come down to sea level to run the marathon, they have an advantage. Why? Because they have more red blood cells. Therefore, they will have more oxygen distributed throughout the body. And oxygen is very important for muscle function and so on. So that's one strategy that athletes use. So the factor here is erythropoietin, that hormone. So you could say, why don't the athletes just inject erythropoietin into their body? Well, that's illegal. You cannot do that. But you can train at high altitudes and have an increase in red blood cells naturally, not by injecting this hormone. Okay, so what are some of the internal stimuli that can disrupt homeostasis? So low blood glucose. So if you have low levels of glucose, if you're starving, and especially the brain is very, very sensitive to low blood glucose. The brain is one of the organs in our body that is more active. And the more active, the more glucose it requires. Why? Because glucose is used to produce ATP, which is the energy source in our body. So if the levels of glucose are very low, then the brain doesn't function properly because it doesn't have enough ATP being made. Also, increased acidity. That's not a good thing to happen. So that can also disrupt homeostasis, in this case, the pH. So most of the conditions in the body have a neutral pH. So if you have too much acid, then the pH is disturbed. So here you have this concept of homeostasis being disturbed by different types of stimuli, different types of stress conditions, some occurring outside the body, so they are external, and others occur inside the body, and those are internal. So, I forget to read here. Oh my gosh, that's really bad. What is the interstitial fluid composed of? The interstitial fluid is composed of water, all kinds of nutrients, oxygen, carbon dioxide, and so on. Oh, that's fine. What are the pores called again? The water pores are called aquaporins. Why are the ions not transported freely throughout the membrane? That's a good question that you will learn. Because these ions, sodium and potassium, are very important for the nervous system to act, for that electrical current that allows the information, the message going from the receptors to the central nervous system. They rely on sodium and potassium. So those ions, the concentration of those ions is highly, highly regulated. So they cannot just move freely. Our anatomy professor mentioned how if water enters a blood cell, it explodes. Okay, so the movement of water is highly regulated, as you will learn, and it's balanced, right? You cannot have more water in the red blood cell and less water in the plasma. The level of water has to be maintained, has to be in equilibrium, because if not, the red blood cells will burst. If there's too little water in the red blood cells, they shrink, they crenate. So the level of water has to be equilibrated. How can sodium pass through the blood vessels freely if the blood vessel is made up of cells? Right, so that's a very good question, but these ions first have to go through the membrane of the cells, the endothelial cells that make up the walls of the plasma of the blood vessels. So all this is highly regulated. Also, some blood vessels, the cells are what we call fenestrated. That means that there are pores between two cells, two adjacent cells. So small molecules and ions can cross through here. But all this process is very complex. I'm just giving you a very general concept. I like your questions. They're all very good questions. And that's what you're here for, is to learn how are the passage of water, these ions and nutrients, regulated from these, let's say, environments, these three environments, from the inside of the cell, the intracellular fluid, through the interstitial fluid, and through the plasma that is located in the blood vessels. So how is all this regulated? And that's the beauty of our body. We don't have to be aware every single second. We don't have to control this. There's mechanisms that keep this regulated. Well, you're asking, I already adjusted the chat. So right now, can everybody see the messages?
Professor, my chat is open, but I don't see others' questions. Like where you are reading the questions and answer, I don't see them.
No, I adjusted it for everyone.
When
It,
I look at
it
the
only
chat,
it
it
only
says
appears
it's
that saying direct message.
Yeah, it's private.
Okay,
It's
so maybe I have to adjust it in the general when I turn off the Zoom. Maybe I can correct it there, but right now there's nothing I can
Yeah.
do. I
Okay.
keep putting everyone in the meeting, but then a name pops up. So I will read the questions, okay? Because right now I don't want to turn off them. Oh, can you mute yourself, please? Mute. Oh, okay. How can high acidity affect the body? Well, because the pH of the body has to be maintained at a neutral level. For example, in the stomach, the pH is very acidic. Our cells produce hydrochloric acid. If you work in the chemical lab and you would put a hydrochloric acid on your skin, it would burn. But to think about it, our own body inside the stomach produces hydrochloric acid. And it's very interesting because why would we need hydrochloric acid in the stomach? Because under acidic conditions, big molecules such as proteins, DNA, and carbohydrates, to name a few, they open up their structure because they're highly folded. They open up so that the enzymes can chew them up into very small molecules that can cross the walls of the blood vessels. So it helps digestion. Having an acidic pH in the stomach, the function is to have to help digestion. In-person exams will be held the same time the class, yes. So do this process count as involuntary? Yes. For example, we cannot tell our body now, stop allowing water to go into the cells in the kidney. No, it's all involuntary. And there's the peripheral nervous system is the one that regulates all these involuntary processes. So it's under involuntary control. So I'm going to read the message, the questions. And I will change this for next class. Let's see.
Excuse me.
Okay. The red blood
So
cells, the correlation to sickle cell disease, yes, because the microbe that called, so sickle cell disease means that the hemoglobin inside the red blood cells, which is responsible for carrying oxygen, the hemoglobin becomes crystallized. And therefore it cannot transport oxygen efficiently. Okay, so when an ion like sodium in the picture leaves the cell, does a new ion like potassium have to enter, restore it? So interestingly, the concentration of sodium inside the cell is very different from the concentration of sodium outside the cell. So there's more sodium outside cells and more potassium inside. There's no equilibrium on the two sides in the cell and outside the cell. The concentrations of sodium and potassium are very different. And they're regulated by these channels that I mentioned, the sodium channel and the potassium channel. So the concentration of these ions outside and inside the cell, these specific ions, sodium and potassium, and also calcium, the concentration inside and outside of the cells are not the same. When an ion, okay.
So Professor, when the potassium is going into the cell, then the sodium is coming out. Do they go at the same time?
Well, you know, there's a protein that's called the sodium potassium pump. And what it does, because it maintains this equilibrium of between sodium and potassium inside and outside of the cell. So this protein is a pump that the sodium and potassium fit very nicely in the pockets of these proteins. Its function is to bring sodium out and bring potassium in. So it's responsible for maintaining high sodium concentration outside and high potassium concentration inside. And, these are all, as I said, you're asking very good questions. And this interplay between sodium and potassium is very, very important for the nervous system to work. And you will learn this in great detail. So if there's a high concentration of sodium inside, then the nervous system doesn't work. How does hypoglycemia affect the brain, given that the brain needs sugar to function? Well, hypoglycemia, you can faint if you don't have enough sugar, if you don't have enough glucose being delivered to the brain, because the brain is so active. So what is interesting is, for example, the muscles, they can store glucose in a form that's known as glycogen. So glycogen is a molecule that is made up of thousands and thousands of glucoses attached to each other. It's what we call a polymer of glucose. So the muscles can store glucose, but the brain cannot store glucose. So the brain has to be constantly fed glucose from the blood. So if there's not enough glucose in the blood because someone is fasting, the level of fasting has to be such that there's enough glucose being delivered to the brain. If not, you can faint and other problems can arise. So sodium cannot cross for the capillary. The movement of sodium is always highly regulated. So the pumps work against concentration gradients, yes, and that's known as active transport, where you will learn. So active transport requires a lot of energy, and what it does, it moves ions against their concentration gradient. So when they go from low concentration to high concentration, they require a lot of energy. So how does sodium get into the cell if the protein is pumping out? Because there's channels that open, there's specific sodium channels that open under highly regulated conditions. So when they open, they allow sodium to get into the cell. But then that pump works in the opposite direction. It pumps sodium back out. So these channels, the sodium channel and the sodium potassium pump, work in opposite directions to maintain the proper concentration of sodium outside the cell and inside the cell. The same for potassium. Does this have to do with homeostasis, or is it a different topic? All this has to do with homeostasis. Okay, so let's move on. So the two systems in our body that are directly responsible for maintaining homeostasis, remember I already told you this, are the nervous system and the endocrine system. Here you see sort of a very simplified view of the nervous system. So in red, the brain and the spinal cord, those are, oh my gosh, what's the time? You are asking such good questions. Those are part of the central nervous system. And then in blue is represented the peripheral nervous system. So you can see that the whole body is wired with these neurons, these nerve cells that can bring messages from the periphery, that's the peripheral nervous system, that is connected to the receptors. So this allows for messages to being delivered from the periphery, receptors that are on the body, they initiate this message that travels from the periphery to the central nervous system. And the central nervous system is the one that is the control center that makes the decision. What is the response that is required to maintain homeostasis? So this is the nervous system. Because it relies on this electrical current that is conducted, that is generated by the movement of this sodium and potassium that I was telling you, the response is very, very fast. But also the endocrine system is responsible for maintaining homeostasis, but the speed of response is much slower. Why? Because it depends on the release of hormones, and the hormones travel through the blood until they reach the target organ. So this is the response of the nervous system is very fast. The endocrine system is slower, but it stays for longer. So again, here you see now the homeostasis, how important it is, and how this feedback loop, we're going to talk about feedback. So I want you to understand the concept of feedback. The feedback is this loop that is generated by activation of the receptors, and it's conducted by that electrical current, generates a response that brings the body back to homeostasis. So this regulation involves, this feedback involves a receptor, the control center, and the effector. So the receptors are distributed throughout the body. You have temperature receptors. You have so the receptors, what is the concept of a receptor? A receptor is a protein, and this protein is highly folded. But when it responds to different stimuli, so a protein that makes up the receptor that is sensitive to heat, the folding of that receptor changes when the temperature increases. The protein that is responsible for the cold receptor, it doesn't respond to a temperature that is increased. It responds when the temperature is decreased. Then you have the ear. The ear is a receptor for sound. You have the eye. The eye has receptors for light. So all these receptors are highly, highly specific for different stimuli, and they're distributed throughout the body. So the regulation is important to maintain homeostasis. So once the receptor is activated, it sends a message through the nervous system, right? Through these wires that make up the peripheral nervous system. This message that is induced by the receptor is conducted to the control center. The control center is located in the central nervous system, the brain and the spinal cord. So the message goes from the receptor to the control center. We say this is the afferent or the input pathway going from the receptor to the control center. So afferent, it's the input. Then the control center decides what type of response is required and sends that message out to the effector. What is the effector? The effector is muscles and glands. You're not seeing the PowerPoint?
Yes, we can see
Can
it.
you tell me if you're seeing the PowerPoint slides?
We can
We
see it.
can see it. We
Yeah.
can see it.
Yes, we see it.
Yeah, we see it.
Okay. The text
They're also
couldn't-
on bright space.
Yeah.
You're
Okay.
in bright space, yeah.
Okay, good. Okay, we are okay, so here we have then the control center sends a response out to the periphery. To where? To muscles and glands. So muscles and glands are the effectors. They receive the message from the control center. So that is the efferent pathway or the output. So afferent goes from the receptor to the control center, and the efferent goes from the control center to the effectors. Okay, so let's look at an example of this circuit. So you have a receptor, this, let's say, a pain receptor. A pain receptor is a very simple receptor. It's just the ends of a nervous system, of a neuron, of a sensory neuron. So you get pricked with a pin, and the pain receptor, if it's strong enough, the pain receptor is activated, and sends a message to the sensory nerve, here represented in blue, so that electrical current flows very, very quickly to the spinal cord. So the spinal cord, this is a cross section of the spinal cord. That's the control center. So you see now the message, that electrical message input goes through the spinal cord. The spinal cord, it goes through an interneuron, and then it activates the message that goes out through the motor neuron. The motor neuron then brings the message to the muscle, so then the finger is removed from the pin. So this happens very, very quickly, in milliseconds. So this is a very short circuit, but you can see that homeostasis is disrupted because the pain receptor is activated. The message reaches the control center, and then the control center sends a message out to the effector. In this case, the effector is the muscle. So you see a very, very simple circuit to maintain homeostasis. Not all receptors are for maintaining homeostasis, like taste, or do they do so in like good, bad taste? Well, these are also receptors that they actually bring, you know, that some receptors can feel bitter or can feel acid and so on. Yes, I would say that this is not homeostasis, but the circuitry is the same. The afferent signals are also sent to the brain, or is it just the spinal cord? The brain and the spinal cord. Mostly the spinal cord, and then the spinal cord sends it to the brain. Okay, so now we're talking about this feedback. Remember, we talked here about the feedback loop, this feedback loop that starts with the receptor going to the control center and then to the effector. So there are two types of feedbacks, a negative feedback and a positive feedback. So what is an interneuron? Interneuron is a neuron that is located just in the central nervous system, and it makes the connection between the sensory neuron and the motor neuron. So remember, all this is an electrical current that is traveling. So the interneuron makes the connection between those two neurons. Okay, so there's two feedbacks, the negative feedback and the positive feedback. The negative feedback is a feedback that reverses the original stimulus. So let me give you an example. If there's an increase in the temperature, then the negative feedback is activated, and the result is to bring down the temperature to homeostatic levels. So it reverses the effect of the original stimulus. A positive feedback enhances, increases the effect of the original stimulus. And you understand this better with examples. So, do the nervous and endocrine system operate independently? Well, not really, because the endocrine system is going to receive messages from the nervous system. So there's a very big interaction. Okay. Okay, so I'm going to give you examples of the negative feedback and the positive feedback. Okay, first let's start with the negative feedback. So let's say you have a viral infection that increases body temperature. So there's receptors that will perceive this change in temperature. The receptors are in the skin and in this area of the brain that's known as the hypothalamus. So these receptors are activated and they send the message to the brain. Now, in the brain, this hypothalamus that also has receptors for changes in temperature, it's considered the thermostat of the body. It's the hypothalamus that controls, in a way, body temperature. So if you have a rise, an increase in body temperature above the normal. So look here at this graph. This graph shows what a normal body temperature should be. So the range is from 37.2 to 36.7 degrees centigrade. So it's a very, very narrow range. If it goes above or below, that means that your body is not healthy anymore. So these receptors are activated when the body temperature goes above or below this range. So then these receptors are activated and send a message to the hypothalamus that it's an area in the brain that is considered the thermostat of the body. So the hypothalamus now is the control center, and now it's going to send a message that is going to overcome that increase in body temperature. So where does that message go? That message goes to glands in the skin, the sweat glands. So the sweat glands now allow evaporation, you transpire, and that evaporation of the water, of the sweat, causes a decrease in body temperature. Also, it causes the blood vessels to dilate. The blood vessels in the skin, they increase in diameter. So they get closer to the surface of the skin and they will lose temperature by radiation. So you see here that the hypothalamus, the control center, when it perceives that there's a change, there's an increase in temperature, immediately it sends a message to the sweat glands in the skin. So they produce sweat and the evaporation of the water at the skin decreases temperature. And also to the blood vessels at the skin, they dilate, the diameter increases so that they're closer to the surface of the skin and heat is lost by evaporation. So these two mechanisms work together to bring the temperature to homeostatic levels. So you start with an increase in temperature and you end up with a decrease in temperature to go back to homeostatic levels. Okay, so I'm giving more examples. Yes, so insulin also works through a negative feedback loop.
Can you explain why this would be considered negative instead of positive?
You mean this circuit here?
Yes, like why is it
because
negative?
it's activated by an increase in temperature, correct? So you have fever,
Correct.
so this you have an increase in temperature, therefore this is what activates this circuit. But what is the mechanism? The mechanism is to decrease temperature, so it goes back to normal. So you have an increase in temperature that activates this circuit, and the result, what the hypothalamus does is decrease temperature. So it's the opposite. It's activated by an increase in temperature, and it results in the decrease in temperature to bring it back to normal. Is it clear?
Yes, that makes sense.
And that's why it's a negative
the
feedback.
temperature rising, which isn't
Right.
good.
So it causes the opposite
Right.
of what causes this circuitry.
Makes sense.
How does the hypothalamus knows that the person has a fever? Like, how does the signal begin? It's not a silly question. It's a very good question. Why? Because these receptors, the temperature receptors, they are proteins, right? So they're folded in a certain way. If there's an increase in temperature, they open up. So it's that change in temperature causes the protein to unfold. The structure of the protein changes, and that's how that receptor is activated. It depends on the change in structure of the protein. I hope that is clear. Well, the lower temperature, the receptor is activated by low temperatures. So the receptors are very specific. There's some for high temperature and others for low temperature. So when you put your hand on the oven, the receptors for high temperature are activated. If you put your hand on ice, then the cold receptors are activated. So these receptors are proteins, the structure of which changes depending on the stimulus. It's so radiation is a form of losing heat. So if you think about when you have a heater in your house, the heat comes out and spreads through, comes out of the heater by radiation. The heat is controlled, is spread by radiation. Does the body react to pain as a negative feedback? Well, there's no negative feedback for pain. So actually there is, because there are endorphins, which are hormones that are released when there's pain. So, you know, when marathon runners, they run for a long, long time. And when they stop, you say that they are high. Why? Because the body secretes endorphins, which are opioids, our own opioids, that counteract pain. So you could consider that. Something that changes homeostasis, considered a negative. So there's negative and there's positive feedback. I'm going to give examples of positive feedback. So having a fever initiates a negative feedback. Would shivering in cold temperatures be considered a negative feedback? Well, shivering increases the temperature. Yes. So you are at a very low temperature, you start shivering, you generate heat. So it is also a negative feedback. Very good. I'm assuming when the hypothalamus detects a lower temperature, we will begin to shiver. Exactly. Do feedback systems only refer to continuous changes in stimuli? Well, the stimuli are not continuous. They stop. This feedback stops, and I'll explain how does it stop. Can a negative feedback also relate to allergic reactions and when you have an EpiPen? Okay, so now these are allergic reactions or reactions to chemicals that that are not natural or not present in the body. So some people can be more sensitive to some chemicals than others, but so a negative feedback. So let me think about, you're asking very good questions. So the cell, when there is, for example, a bacterium or a microbe or a chemical that can be harmful, some cells become activated and start releasing chemicals that will kill bacteria. That's the whole principle of the immune system. So you could, in a way, consider it as a negative feedback. If a receptor is responding to much, let's say insulin, is lowering the sugar too much, will the body send hormones to stop? Yes. So there's insulin and there's glucagon. So these two hormones that are produced by the liver, they act in different conditions. Insulin to increase the glucose coming into the body, and glucagon has the opposite effect. So when temperature decreases is a negative feedback. No. Temperature decreases or temperature increase, they both generate a negative feedback, because what happens is that the body will generate a response that will counteract the increase in temperature or the decrease in temperature. So if you have an increase in temperature, the body will generate a response that will decrease the temperature. If you have a decrease in temperature, the body generates a response that will increase. So these are all examples of negative feedback. Okay, we get into the childbirth, which is a positive feedback. Okay, now let me go a little further, because if not, we'll never get out of here. Okay, so negative feedback. I think this will answer some of your questions. So here's the negative feedback. So you have initial stimulus, and then you have the response, and then you have the stop or the response that will overcome the effect of the initial stimulus. So once homeostasis is reached, that feedback is stopped. So it starts when homeostasis is disrupted, and then the message is sent to the control center, and then the response is generated. Once it reaches homeostatic conditions, it stops. So this negative feedback doesn't go on and on forever. If it would go on and on forever, it could lead to disease. So negative feedback stops when homeostasis is reached. Okay, now let's give examples of positive feedback. So let's say you have a cut on your skin, and you cut the blood vessel, and blood starts leaking. So how does it stop? This is a very dangerous condition. You don't want blood to leak for a long time. So what happens? In this case, you have platelets. Platelets come together to form a clot. And why is it a positive feedback? Because these platelets, these initial platelets that arrive at the site of damage, they will release chemicals. And these chemicals are known as chemoattractants. What does that mean? They attract other platelets. So there are chemicals, the platelets release chemicals that will attract more platelets. So you get more and more and more platelets that come together and form the clot to prevent leakage of the blood. So this is a positive feedback, where you have a cut in the blood, you have platelets that release chemoattractants, and this will bring more and more and more platelets to the site. So it's enhancing the effect. So it's a positive feedback. Also childbirth, someone asked about childbirth. So what happens, you have in the uterus, they are stretch receptors. And when the head of the child reaches the uterus, the muscles will stretch, and these stretch receptors are activated. They send a message again to the hypothalamus. You will learn that the hypothalamus is a very, very important brain area that controls many, many functions. So we talked about how the hypothalamus controls temperature. Now we're saying here that the hypothalamus plays a very important role in childbirth. So you have the uterus contraction, you have the stretch receptors being activated, send a message, the afferent message, the input to the hypothalamus. And now what happens? The hypothalamus secretes this hormone known as oxytocin. And what does oxytocin do? Now remember, this is a hormone. It's released into the blood, and it travels until it reaches the uterine muscles, so the uterus, the muscles in the uterus. And what happens? It induces further contraction. So you start with slight contractions when the head reaches the uterus. So you have contractions being initiated, the message reached the hypothalamus. The hypothalamus now releases oxytocin, which induces more contraction. So here you again enhancing the effect of the stimulus. You get having more and more and more contraction. This is known as the Ferguson reflex. So I gave you two examples. Blood clotting and childbirth are examples of positive feedback. So let's see, where did I stop? So would oxytocin be released during childbirth be considered positive? Yes. Why do I have two opposing negative feet? Sweat from my armpits when I am shivering. Well, you don't shiver and sweat at the same time. Sweat is to lower the body temperature, and shivering is to increase the body temperature. Is the production of androgens and sex hormones an example of positive feedback? Well, I'm not sure what you mean here. Can some of, because the sex hormones are involved in puberty and so on, can some of these systems fail? Yes. And that leads to disease. When you cannot maintain homeostasis, when these feedback loops are not working, then this leads to disease. Do antidepressants reacting to the brain count as a negative feedback? No, not everything is a feedback. There are many functions of chemicals in our body that are not activated by feedback stimuli. Not everything is feedback. Is an autoimmune disorder also harmful feedback? No, as I said, not everything is feedback. It's based on the feedback loop. Yes, so the negative is basically the opposite. So yes, the mother, the birth of a child and the dilation is a positive feedback. When people are induced into labor by doctors, do they add a chemical? Yes, actually, how the labor is induced, many times oxytocin is involved. So a positive feedback enhances the effect of the stimulus. The negative feedback opposes the effect of the stimulus. Okay, so someone asked, what if these feedbacks don't work well? For example, what stops? So we know that the negative feedback stops when homeostatic conditions are reached. What about the positive feedback? When does it stop? The positive feedback needs an external event for it to stop. For example, in childbirth, when the child is born, then that positive feedback ends. In the platelet, when the clot is formed, then the positive feedback stops. So you need an event, an external event to stop the positive feedback. And what happens if, for example, if there is an increase in body temperature, if it reaches very high levels, for example, if it goes to 104 degrees, this high temperature will increase a lot of the metabolic reactions that occur in the body. And these metabolic reactions will increase temperature. So here, the negative feedback is not happening. You have actually a positive feedback because you have a high increase in temperature that increases metabolic reactions that will produce more heat. This is why when there's an increase, a high increase in body temperature, sometimes you are advised to take a cold bath or cold shower to lower the temperature because the negative feedback is not enough to lower the body temperature. Okay, so now we reach the homeostatic imbalance, which leads to disease. And how can we define disease? How can we know that there is a disruption of homeostatic balance. So there are signs that can be measured. You can measure fever, you can measure blood pressure, and so on. These are measurable signs. They're objective. It's not like asking the patient, do you have a pain? And that's symptoms. So the difference between signs and symptoms caused by disruption of the homeostasis is that signs are measurable, they're objective. Symptoms are described by the patient if they have headache, for example. And then you have diagnosis of the disease. What kind of disease does the patient have? Then you use tools and tests that can lead to the diagnosis of the disease. Okay, so let me give you examples of tests that can be used to diagnose a disease, because although you have a disruption of homeostasis, it can occur many different causes. So the treatment depends on what stimulus, what condition caused the disruption of homeostasis. So for example, very interesting tool that was recently, or test that was recently developed, is this Biol screen application. So pancreatic cancer is very, very hard to diagnose. When it is diagnosed, it's already usually too far advanced. So this test bases on the color of the sclera, the white of the eye. So there's this pigment that is produced by the liver, and it's yellowish. The color of bilirubin is yellowish, and it is goes from the liver to the pancreas. And if there's a cancer that impedes the movement of bilirubin from the liver to the pancreas, then it accumulates and causes the skin to become yellowish and the sclera, the white of the eye, to become yellowish. So this test, this application, uses the iPhone together with these glasses that can assess the yellowish color of the sclera when it's not visible with eyes that are not protected. So it's a very sensitive test that can lead to diagnosis of pancreatic cancer. Now, don't think that someone that has a yellowish sclera does not mean that they have pancreatic cancer. It can be caused by many other conditions, hepatitis, and so on. Okay, so I want to get to the end of this. So I'm not going to answer questions anymore. I want to get to the end. So let me describe some other diagnostic tools. So you have X-rays that are based on solid parts of our body that will not allow the X-ray to go through, and therefore you have images, an image is generated. So for example, bone is highly dense and doesn't allow the X-rays to go through, and therefore it generates an image on the film. So it's also used for mammography. Then you have CAT scans or computerized tomography. They're also based on X-rays, but the advantage is that they take X-rays all around the body. So they give you a 3D vision of dense parts in the body, tumors and so on. So the advantage is to give a 3. It doesn't give you a color view. Of course, it's just like an X-ray, but because it is the X-ray is taken all around the body, it can give you a 3D view of cancer and so on. Then you have DSA, or a digital subtraction angiography. And this is very useful for studying blood vessels. So a patient takes an X-ray before and after the patient is injected with an opaque dye. So you take an X-ray before and then after, and you can see because the dye is opaque and it flows through the blood vessels, it gives you a good view of the blood vessels if there is an aneurysm or something like that. So this is angiography. It's used to study blood vessels. Then you have positron emission tomography. This is a PET scan. And here, you're not looking for an image. You're looking for a change in function. So what is the idea here? You inject the patient with a radioactive compound, such as, for example, glucose. So this radioactive glucose is taken up by cells, and cells that are very active, such as cancer cells, will take up more of this radioactive glucose than normal cells. So then if you take an X-ray of this individual that has this that was injected with this radioactive dye, it will give you a view, an image that shows where in the body this radioactive glucose was taken up, which cells took up higher amounts of this radioactive glucose. And if there's more glucose being taken than in other areas of the body, this can give you an idea if there's cancer in those areas. Why? Because cancer cells are very, very active and they will take up more glucose than normal cells. Then you have magnetic resonance imaging, and this is used to view parts of the body like ligaments that are not opaque. So you take X-rays to see the bone, if there's fractures in the bone. But for example, if there's problems with ligaments, the X-rays cannot be used because the ligaments are not opaque enough. So you use a magnetic resonance imaging that is based on a very, very strong magnet. It's 3,000 times stronger than the magnet in the earth.
Professor,
So this,
515.
yes? What?
I mean, it's above 515. It's 519.
I know, but I'm going to finish this, and it's going, if you have to leave, leave. It's going to be recorded, right? So this MRI, magnetic resonance imaging, is used to image soft tissues, such as ligaments. And this is the last slide, just one more slide. This is ultrasound. Oh, so if someone is pregnant, they cannot use a magnetic MRIs because it might affect the fetus. The magnet is so strong. So then you can use a sonogram. This takes advantage of sound waves. And it's good to view live and moving parts of the body, such as a fetus or such as the heart, the pumping of the heart. So you take advantage of sound waves to get images of parts of the body that are moving. Okay, so we went through disruption of homeostasis until our maintenance of homeostasis, disruption of homeostasis, and now how can you diagnose the disease state using these tools? Now, the last thing I want to say is the organization that we are going to go through all these different levels of our body organization, going from the atom, the simple atom, to molecule, organelle, cell, tissue, organ, and then the whole organism. So an atom is organized into different atoms, make up a molecule. Different molecules make up organelles that are small parts in a cell. Many organelles make up a cell. Many cells of the same type make up tissues, and different tissues make up organs. So, for example, the heart is made up of different tissues, and then different organs make up the organ system. So remember, there's 11 organ systems in our body, and then all the organ systems together make up the organism. So throughout the semester, we're going to go through all these different levels of organization. So I thank you for all the different questions that you asked that were excellent. So, okay, so the class continues, but it is recorded. So those that had to leave early can hear on the recording of the class.
Professor, I have a question.
Yes,
I was just wondering for the lab quizzes, is it just like the studying of anatomy, like the models, like
yes.
from
So for the lab, we have to separate lab from lecture, because if not, I won't have time to go through the lecture. So lab questions, please ask your lab instructor. That's why the lab instructors are there. So you can ask, you have 3 hours and 50 minutes. They have lots of time to answer your questions. Here in the lecture, I will only address lecture questions. Let's be organized, please.
Okay, thank you. Sorry.
Okay? It's not that I don't want to answer, but I just have to keep it organized. Okay?
Professor, I have a question.
Yes.
I see in the lecture note number 2, you have the chemical level, cellular level, tissue level. Those are you gonna discuss next lecture, or we have to read on our own?
I don't understand what you're asking. Can you say it again, please?
So I see in your you posted in the Brightspace the lecture note number 2.
Yes, it has more.
It has more
no,
topics.
just the
Should
slides that we studied today. The extra slides will be discussed in the next lecture.
Okay, thank you, Professor.
Okay, with chemistry.
Okay, thank you, Professor.
Yes, the slides. Actually, I'm going to take them off. The lecture slides.
Is class over now?
Yes.
Can we leave now? Okay.
Yes, you can leave.
Bye-bye.
Have a nice day.
Print something out.
Have a good evening.
Okay, have a good weekend, everybody.
Have a good weekend, Professor.
Have a good weekend.
Bye. Thank you, Professor.
Thank you, Professor.
Bye.
Thank you. Have a good weekend.
Bye. Take care.